Right. Good afternoon, CGIL. This week, we're very happy to welcome back Dr. Pablo Fonseca, a postdoctoral fellow at the University of Lyon. Dr. Fonseca obtained his Bachelor of Science in Biology at the Federal University of Minas Gerais in Brazil. Subsequently, he completed his PhD and master's degree in genetics with an emphasis in genomics and bioinformatics at the same university. From 2017 to 2021, he joined Dr. Canovas here at CGIL as a visiting student in 2017 and later as a postdoc in 2018. Since 2022, he has joined the GALLO group at the University of Lyon as a postdoc through a grant from the European Union for the Attraction of Talents. Today, Dr. Fonseca will be presenting his research entitled Genomic Annotation in Livestock for Positional Candidate LOci, GALLO, Applications, and Updates. So we'll turn it over to you now, Dr. Fonseca.
So thank you very much, Gabriela, for the kind introduction. Nice to see everyone this week again. I hope that you don't get bored with another presentation in a very short period, but I think that today you're going to talk about a different topic. And just to let you know, I'm not seeing the screen with the teams. So if something happened or someone wants to ask a question or whatever during the presentation, please, you can talk because I will not see the screen with the presentation, okay? So just to start, so the idea of this presentation is talk a little bit about GALLO, our package that was developed while I was in Guelph in the group of Dr. Canovas during my postdoc. You're gonna see that the idea of this presentation is not only show the functions that GALLO has, but also try to show some context about how to use the GALLO functions in different contexts. I try to summarize the theory behind some of the mainly questions that I receive about GALLO. So let's see how it's gonna work. And at the end, if you have any additional question about GALLO or anything else, feel free to ask, okay? But GALLO, I used to say that is a side effect of my PhD thesis, where we were trying to integrate different sources of omics data, different results from multiple association studies, and the main idea of these projects were trying to identify common candidate genes in multiple genomic regions. So we were annotating genes, comparing results among studies in a very repetitive way, and I was a little bit tired to do this manually or using the tools that we had at that period. So I decided to create some functions in R, and these functions I start to share with some colleagues, and at the end share the R scripts were a little bit difficult for me, so I compile everything in a package, and at the end we decided to publish this package on CRUN. This took a while, but at the end we had GALLO, that we decided to call the name because it's a common name of a livestock species across the mother tongue of the three countries of the GALLO's authors. There is nothing related with the mascot of the most important soccer team in the world. It's just a coincidence, but you can see there. And since its publication in 2020, GALLO already reached more than 35,000 downloads and 130 citations. It's much more than I was expecting. It's a little bit overwhelming that you think that a lot of people is using the tools that you were developing and maintaining, but it's also very nice. And GALLO was used in different contexts for different species in studies that are published in very important journals. So it's very nice to see how this tool can help different kinds of investigations across the world. And what GALLO tried to provide is a set of tools that allows the user to do accurate annotation of genes and quantitative trait loci, QTLs, that are located with candidate regions. Can be interval, position of a candidate marker. This interval can be, for example, a haplotype, a window, a CNV, whatever. Results basically that you can obtain for the most common genomic and other kind of omics analysis that we do for livestock species, for example, such as GWAS, transcriptomics, but it's not limited only for these two methodologies, okay? The first version of GALLO that was released on CRAN was composed by 8 main functions that were connected in a straightforward way that try to provide, as I mentioned, a set of functions to annotate genes and KTLs that allows also the user to do data comparison, enrichment analysis, and visualization for different kinds of results from association studies. And I believe that the most common one that we are involved with is GWAS. And I will try to use the context of GWAS to try to explain the use of GALLO and the functions of GALLO in the next slides, okay? But just as initial point for this presentation, what is a GWAS? Basically, GWAS is association study, and as any kind of association study, we have a hypothesis that we are testing in a sample, and we try to extrapolate our hypothesis in conclusions that can be applied to the population where we obtain the sample. In the context of GWAS, the hypothesis that we are testing is the association between genetic variants and phenotypes. For this, we need a sample, phenotypes, and genotypes. And I'm sorry, I just realized that I have this figure in Portuguese, not in English. Okay. And so what we want to check, we want to see if there is statistical difference in the phenotypic values of observed in individuals with different genotypes. Okay. And using, for example, the Falconer model for a single biallelic KTLs, if we try to do, for example, linear regression to test the association of different genotypes with our phenotypes, the slope of this regression will account for the additive component of the variation across the genotypes. And what we are really testing if is the slope of this linear regression is different of 0. So if we have a slope that is different of 0, we consider that we have association between a phenotype, between a genotype and a phenotype. And in a GWAS, what we do? We do for each SNP that we have in our panel of SNPs, fitting of a statistical model, and with this we can have a genomic view of the association pattern of the markers across the genome with our phenotype. And in this way, targeting some candidate regions across the genome that might be harboring causal mutations for our phenotype. Okay, but it's very important to know that when you are doing a GWAS, usually, most part of the time, in the recent years, the number of studies that are a little bit different from what I'm going to say here increasing when you go, for example, for whole genome sequencing, but the number of whole genome sequence GWAS is much smaller than the ones using genotyping panels. In the context of genotype panels, what we do, we have some pre-selected markers that we fit in our model and try to find association. These markers, they are selected not, in the majority of the cases, actually, they're not selected by the effect. So we don't know if this marker has a direct effect over a phenotype. They are selected based on representativeness. So to cover different regions in the genome. So they try to be split or distributed evenly across the genome, but they also try to have a good representation of the linkage disequilibrium pattern in specific regions, trying to represent different kinds of haplotypic groups. So we try to cover regions in the genome using a representative SNP for each one of these blocks that we have. So why I'm mentioning this? Because when you do a GWAS, even if you have a significant result, you are not seeing the effect of the causal mutation directly. Most of the times you are seeing an indirect effect of the variant that we have in our SNP, in our SNP panel. So the process of data pruning, the process of functional analysis of our results is extremely important to try to identify the candidate genes and consequently searching for causal mutations that could help us to understand better the biological process associated with the variation in the phenotype and even more effective selection of the animals, because we try to exclude the different effects of the linkage disequilibrium in different populations when we are working directly with the causal mutation. And as I just mentioned, when you do a GWAS, we need to try to split two different contexts, association and causality. Association is when we observe a presence in the change of one variable, and this change in one variable is related with the change in another variable, in a second variable. So they are associated. We can see that, for example, you can say, for example, that they are correlated. But causality suggests a direct influence, okay? So we can have association without causality. And this is really important to keep in mind when you are doing a GWAS, because sometimes it's difficult to interpret the result of a GWAS just because of this. And just to give a very clear example, we can have different kinds of spurious correlations, different kinds of spurious associations. I like a lot this database that shows some a very large range of spurious correlations. For example, here showing the GMO use in soybeans in North Dakota and the correlation with geothermal power generated in Russia. And you can see that it's a pretty much a correlation of one. And these are spurious correlations, of course. But even when you have a spurious correlation, if you push too much, you can try to find an explanation for this. And in this database, it's very nice because they use artificial intelligence to create a scientific paper, fake scientific paper that explain the correlation, the spurious correlation. And you can, if you have curiosity, you can go and read this publication where they force the causality in this spurious correlation. So we need to take care when you are interpreting results of a GWAS, because we know the flow of biological information is a very complex process. Even when we look for the first or the most simple design of the central dogma of molecular biology, we have a lot of process that can be regulated in different levels or can auto-regulated. So we go from the DNA to the protein. In the middle of this process can have a lot of modifications. And you know that the central dogma of molecular biology actually is not so simple and can be expanded in a very complex way, and you have much more complex levels of interactions and regulations. So go from association to the causality, and proof causality is a very complex task when you're doing GWAS. And in order to try to go to the source of, or to prove causality in our results, we need to do GWAS, but we also need to be based in the post-GWAS analysis. When you're going to do the functional analysis, try to narrow down the region, find the candidate causal mutation. And the process of finding mapping, identification of positional candidate genes is really important for this. Finding mapping is the process when you try to narrow down and try to filter our results in a GWAS using, for example, linkages in equilibrium information of our positional candidate genes to try to rank the most probable regions that are harboring our causal mutations. And in order to do this in a very effective way, we need to have knowledge from different areas, so such as population genetics, system biology, quantitative genetics, bioinformatics, statistical modeling. So you can see it's a complex process. And we need to follow the scientific methods. We need to in order to prove association of a sequence of DNA or with a phenotype, we need to do hypothesis checking. We need to follow steps of a scientific method in order to validate our observations. We can not just pretend that we don't know the scientific method and skip directly and say, okay, I found this mutation, and this mutation is the causal of my phenotype. We need to follow a scientific method to prove this. And showing the relationship between genotype and phenotype, as I mentioned, it's not an easy process. It's very complex. It's pretty much like trying to find a needle in a haystack. We don't know in which level of biological information you should look directly, how we can integrate multiple levels of biological information to help us in this process. But of course, the first step is trying to find potential functional candidate genes that have a relationship with our phenotypes and are close from the significant regions in our GWAS that might help us to search for causal mutations in next steps of our research. And this can help us to find the causal mutation, but anyway, it can be a very complex process. But GALLO tried to help us find this needle in a haystack a little bit faster. So it's a very useful tool for that. And how GALLO works. The first thing that we need to have to work with GALLO is an input file. And the input file on GALLO is quite simple. What we need a column with the chromosome and a column with our coordinates if we're working with SNPs. So we have one single column with the position of a SNP in a chromosome. And if we're working with a window, such as a haplotype, a run of homozygosity, a CNV, whatever, we need a column with the chromosome, a column with the start position, a column with the end position, okay? And we can include any additional column that we want in our input file. And this is quite useful because, for example, if you have results from multiple models of your GWAS, you can create additional column with the model name or the population name or the phenotype name and annotate all the candidate genes simultaneously in a single run of GALLO for all your results, and later you can filter the results, the individual results of each model, of each population, just filtering this additional column that you include. For example, here, I have a column with reference or breed. We could annotate all the genes and later filter by breed or by reference, okay? And the first function of GALLO is the function where we import our database. The database that GALLO use can be two different kinds of databases. One with KTLs that must be downloaded from animal KTL database. I receive a lot of emails of people asking why they are not successfully annotating KTLs using GALLO, because they are downloading GFF files from NCBI. NCBI is not a database of KTLs. So the GFF file can be downloaded from NCBI, from an ensemble, but this GFF file will contain gene information. So the GFF file that GALLO used to annotate KTL must be downloaded from animal KTL database. And to annotate genes, GALLO currently uses the standard format of ensemble GTF file. Okay. In future update of GALLO, I'm working to do this process automatically for both NCBI and ensemble, and even maybe for UCSC or other genome browsers, but this is a little bit complex to do this automatically. But for example, you can import the GTF file on R manually, change the name of the columns, and use on GALLO without any problem. And one thing that is really frequently asked to me and for other people that are using GALLO is how work with information from different SNP ships, information about when you are using different populations that are genotyped with the same SNP ship, but the markers have different names, okay? The important thing is that we must standardize our coordinates, and even better if you can standardize our marker names. Okay, so when you have this situation, it's really good. Before you start Gallo analysis, be sure that all your markers are in the same pattern of coordinates and names. The name will not directly affect Gallo, but it will be easier for you to interpret your results later. Now, another very important point, we need to be sure that we are annotating KTLs and genes using the same genome version that we have the coordinates of our markers, okay? So for example, if you have your SNP map on the UMD 3.1 genome version of the bovine genome, and you try to annotate genes using the ARS UCD 2.0 version of the cattle genome, you're gonna have totally different results, okay? For example, here I show the difference in coordinates of the get1 in both of these coordinates genome reference that I mentioned, and you can see that has a substantial change. The genome coordinate is the coordinate of the markers in different reference genome versions. Is not so important when you are doing GWAS, the fitting the statistical model, if you are not consider the proximity or we are not doing sliding windows approach. This will not affect our GWAS results, but the annotation of the genes, yes. So this is really important. We have tools to try to remap this genomic coordinates. We can use the lift over tool on the UCSC genome. We used to have one in the CBI, but is no longer available. So we have tools. You can use BLAST using the sequence close of our SNP to remap in different genome versions. But one very nice way to do this is going to the marker, to the SNP panel information, try to download the manifest file. There you have all the information about this SNP chip. So for example, flanking sequence around the SNP, the reference genome that was used to build the SNP map, the standard SNP map for this SNP chip. And also you can get, if it's available, RSIDs and the name of the markers. And once you have these coordinates or the RSIDs, you can go for a database such as the Ensemble, download the GVF file that has all the RSIDs with the coordinates for all variants that were deposited on Ensemble for a specific version of your reference genome, and you can match the RSIDs and the coordinates across GVF files of different versions of your genome. Okay, so for example, match the coordinates of the markers that you have in your SNP map in the UMD 3.1, get the RSIDs during this match process, download the GVF file from the RSUCD 1.1, and using now the RSIDs that were retrieved from the previous version of the genome, find the new coordinates in the new reference genome. And this you can update and use a new reference genome and search genes in that coordinate, in that version. Once we have the database important on GALLO and also the input file, we can use pretty much the mainly function of GALLO, that's find genes and KTL around markers. I know it's long. I should try to find a shorter name for this function when I first created, but it's what we have now. And what this function receives as parameters, the database that we import, the input file that we create, the method, so gene or KTL, if you want to annotate genes or KTLs, okay. The strategy that you're going to use to retrieve the genes or the KTLs is SNP or haplotype. SNP, if you only have one column with the position of a single, with the coordinate of a single position in the genome. Haplotype, if you have a window with the start and end position, and the interval. So the interval that we want to use to annotate genes upstream and downstream of our coordinates. Okay. This function is based on data tables more specifically in the fourth lapse function. That's a fast binary search based on overlap join, and it's quite fast when you compare the raw results with other tools such as Biomart or bad tools. It's not faster, but there is a reason why it's not faster, and a very important reason. It's because we have all our results in the same environment, in the same output structure, and you don't need to back solve the results to connect markers and genes, for example. For example, when you use Biomart, you inform a list of genomic coordinates, and then you annotate the genes, but then you get a list of genes exclusively. You don't have the associated SNP or the associated region. Using GALLO, you have this. You have the output file of GALLO composed by all the columns that you inform at the beginning, followed by all the information that was retrieved from the database that you were using. And for example, if a single marker is annotated in a region with 5 genes in the proximity of the interval that you use, the row of your input file is going to be repeated 5 times and connected with the information of each gene individually, as we can see here. And this helps us to easily connect a marker with a gene, with a KTL, and then compare, for example, results of multiple models, multiple populations, much easier. Some troubleshootings of this process, as I mentioned. We need to take care if you are using GFF from animal KTL database to annotate KTLs and GTFs to annotate genes. It might seem silly, but I receive a lot of questions about this, okay? Chromosomes must have the same code pattern in the input files in the database. So for example, if you have in the database the chromosomes named as CHR1 and in your input file as 1 till X, whatever, we're not going to find match. So we need to have the same pattern of chromosome naming in both files, okay? And some GFF files in the animal KTL database, they might have the start coordinates larger than the end coordinates. They are switched. And this is because of the remapping process from one genomic version to the other. And we need to switch this before annotate, okay? And we can use on GALLO custom GTF and custom GFF files for any kind of annotation that you want. But it's mandatory that they must have the same column names as the standard GTF and GFFs that we use on GALLO. Okay. But one very common question that I receive in the process of GALLO using by the other users is, which interval should I use to annotate my genes, my KTLs in my GWAS results? This 50 KB, 100 KB. It's a not very simple question to answer, but we can try to find some help to define this region. Okay, we need to take care about. We need to pay attention about what I mentioned before. That is how the platform that we are using was built. So when we use a much denser SNP panel with a larger number of markers, this will affect the length of the interval that we can use. If we are using a custom SNP panel that have causal mutations there, this also will affect. But if we are using standard genotyping panels, we can try to base our decision on the linkage disequilibrium pattern, for example. And we know that the linkage disequilibrium tries to show the association between alleles in different loci. So we try to use the linkage disequilibrium as a measure of correlation in the segregation of these two alleles in different loci. So you can see how frequently they are segregating together. And they will segregate in what we call the haplotype blocks that are delimited by the recombination between 2 points in the genome. And as I mentioned, the density of our SNP panel might affect our capability to estimate this linkage disequilibrium and also to identify this possible haplotype block. So we need to take care about this, okay? And we need to know as well that usually we have a clear pattern of descending clearly descending pattern in the average linkage disequilibrium when we increase the distance between two markers in the genome. Okay, the larger the distance, usually smaller is the linkage disequilibrium because we have more recombination points across between these two SNPs, and then we break the segments of the chromosome, and then we increase the independent segregation of these two chromosomes, reducing the linkage disequilibrium. Okay. And when we do a GWAS with a standard SNP panel, the most common association that we identify is the indirect association. So we see association of a SNP that is in the SNP panel with linkage disequilibrium with our causal mutation, and then we detect this association. A direct association could be, for example, a mutation that we know that change function of a gene, and this function of the gene directly affects our phenotype. But we cannot, usually we don't see these results directly in GWAS. If we're doing, for example, GWAS with a whole genome sequencing, this might happen, okay? So, the LD pattern, the linkage disequilibrium pattern, might help us to delimitate these regions that we want to search for the causal variants. And this linkage disequilibrium and this indirect association pattern actually is the cause of the pattern that we see in the Manhattan plots that are usually used to visualize GWAS results. This kind of pattern of increasing significance in a region that is significantly associated with our phenotype usually is a reflect of the linkage disequilibrium pattern in the region close of our causal mutation. And this is caused by this representativeness, selection by representativeness that we have in the SNP panels. The increase in the significance tends to follow the increase in the proximity of the SNPs that we have in the genotyping panel and the causal mutation. Based on this, if you check the pattern of decreasing the average linkage disequilibrium in different distance across the genome in multiple species, we can see that usually high linkage disequilibrium average, actually high average linkage disequilibrium can see in distance up to 50, 100 kb between two markers, okay? So usually by average, if you try to keep an annotation interval of up to 100 kb, you can assume that you are covering the highest linkage disequilibrium average, okay? But this will not solve all your problems. Why? Because the linkage disequilibrium pattern is not uniform distributed across the genome. We have hotspots of recombination, cold spots of recombination. We have different patterns of association of two markers that are segregating in the genome. So this might affect the length of the haplotype blocks. This might affect how well we're going to annotate a causal mutation. Okay, you can have, for example, a causal mutation in a silencer, in an enhancer that is a distal regulatory element, and this will affect the function of a gene that is 1 megabase, 2 megabase far away from your candidate regions. So we need to take care about this, and you need to consider this when you are annotating candidate genes. An alternative approach, instead of use intervals, should be use the haplotype blocks, so you can estimate the whole haplotype blocks that you have across the genome, identifying which haplotype block you have your candidate regions of the GWAS, and annotate the genes within these haplotype blocks. Once you have the coordinates of the haplotype blocks, you can use GALLO as well to annotate the genes using an interval of 0 and just using the coordinates of the haplotype blocks. Could be alternative approach. Much more complex and difficult, but it's alternative, okay? GALLO has also visualization functions that I'm gonna talk very fast today, but you can check, for example, results across different studies, populations, and summarize these using overlapping matrices. These overlapping matrices can also feed another function of GALLO that's gonna create for us heat maps that show in a very graphical way the overlapping between groups, and then you can see if you have a specific phenotype that share more positional candidate genes or KTLs with other phenotypes. We can summarize the information of the KTLs that we annotated in our candidate regions using the plot KTL info. You can do a pie plot that shows the frequency of each KTL type that we have in the animal KTL database. And you can also do a bar plot that's going to do a zooming in each slice of this pie plot and show the composition of the frequency that we're observing in the slice of the pie plot by then each KTL name that we have within each KTL type. So for example, we have here 15% of reproduction KTLs in the pie plot. If you do a zoom in, we can see that Calvin Easy accounts for approximately 3.5% of this slice, okay? And I think the function that in my opinion, is quite useful in GALLO is the KTL enrichment, the function that we do KTL enrichment analysis. And we know that when you are annotating KTLs or annotating any kind of information in any database, the simple bias of investigation can affect our results. And we know animal KTL database and any kind of KTL database has a strong bias of investigation. We know that there are some sources of phenotypes that are much more analyzed, that are more important. So we have more association studies, we have more data available in this database. And this can be a clear example for if you focus on milk-related traits in the animal KTL database for cattle. We have a much larger number of GWAS in this GWAS or any kind of association study in this database. And we can have an even more specific bias in this database when you go, for example, in the distribution of the KTLs across the chromosomes. When you focus on milk-related KTLs, you know that the chromosome 14, where we have the DGAT KTL, has a very strong effect for milk-related traits, and you can have a bias of results on this chromosome, and this might affect your annotation. And one way to be sure that we are not observing a result of a KTL annotation by chance, and we are really annotating some specific kind of KTL more frequently than we would expect by chance, is doing an enrichment analysis. And the KTL enrich function GALLO does exactly this. So we inform to this function the results of our annotation and also the database that we use to annotate the KTLs. And we can do the enrichment analysis by the KTL type. So as I mentioned, reproduction, milk, carcass and meat, health, the KTL types that we have in the animal KTL database, or for each KTL specifically. For example, milk yield, stillbirth, feed efficiency, the KTL names that we have there. And we can do also this analysis by genome-wide or by chromosome, okay? And we can set all this by the parameters that we have. And this function pretty much is based on a hypergeometric test. And I'm not going to focus on this today because of the time that we have, but pretty much what a hypergeometric test does is try to estimate the probability to observe a result that a number of success that in a drawn process more frequently than we were observing. So this is pretty much based in some parameters. So the number of success, the number of classes that we have in our background, the number of the classes that we are notating in this background. So pretty much what we're doing, we are trying to see if the number of KTLs of a specific class that we annotated in our process is expected to be found by chance or not if we draw the same number of KTLs randomly from this database, okay? And based on these parameters, the background that we use might affect our results. So it's really important to pay attention in the constitution of the KTL database. So if we're using for the KTL database of some livestock species, that the number of association studies is very low, this will affect the bias of the database, as I mentioned, might affect as well the quality of the annotation. So we need to try to pay attention and try to filter the database by association studies that we know that were well conducted. The statistical model can affect also our enrichment analysis. In the case of GALLO, we are using only, as I mentioned here, hypergeometric test. And we need to apply multiple testing correction. But of course, another thing that's really important, maybe we should not focus only on significant results in this enrichment analysis. Sometimes we don't need to focus on enrichment analysis. We just want to see the association, the overlapping patterns across our candidate regions and the KTL that was previously annotated, okay? And of course, results of KTL enrichment analysis. This is just an example of the output of KTL enrichment analysis on GALLO. But what I want in this a way to visualize, but as I mentioned, I'm not going to talk a lot about this, but I want to focus on this. The results of KTL enrichment as any other kind of enrichment analysis for gene ontology, mesh terms, pathways, must be carefully interpreted, okay? Because what we are testing is enrichment in function of a database. So we are seeing if a KTL is observed in our results more frequently than we would expect based on the frequency that we have this KTL annotated in the database. So if a phenotype is very here to be analyzed, so if we have very few KTLs annotated in the database, this might affect our results. Also, if you have the totally opposite pattern, a phenotype that's very frequently affected. So this must be considered, okay? And so the results might be affected by the trait and the species, as I mentioned, if you have a lot of studies or not, and consequently must be carefully interpreted. So enrichment analysis is always is not the final result. We need to interpret the result as well. So we cannot close and finish our discussion or just mentioning a result of enrichment analysis. It's a result as any other kind of association test that we're doing. You need to go deeper in this analysis and try to understand what we are really observing. Relationship plot is another function of GALLO that we can see the connections between SNP and the genes that were annotated, SNP and KTLs that were annotated in our candidate regions. Use this kind of chord plots that show in a very graphical way the connection between the points, but might be tricky when you have a lot of markers and a lot of candidate regions or KTLs annotated. So this can be very messy to see. And one of the reasons why you try to improve GALLO for a version 1.5 that is currently available on Chrome was trying to improve these visualization tools, but also providing other functions. But I'm putting here that I'm showing that maybe soon we're going to have GALLO 2.0, and I'm going to say to you why 2.0. Just summarizing, version 1.5 of GALLO was updated with 6 new functions, and I'm going to shortly mention the six new functions. The first one, we implement the methodology of Bolomar et al. 2014 to estimate pleiotropic effect. In my last presentation, I talked a lot about pleiotropic effect. So I'm going to skip this today, but we know that based on the Bolomar et al. Methodology, we can estimate pleiotropic effect using the summary test statistics of multiple GWAS. So we are going to provide to GALLO the t-test, the test statistics for each marker, for each phenotype that we analyzed in a matrix format, n by m, n number of SNPs, m number of phenotypes, okay? And using a chi-square distribution with the degrees of freedom equal the number of phenotypes that we have, we can estimate p-values for the pleiotropic effect of these markers, okay? Just to mention, it's a very efficient approach. This multi-trait test statistics is quite useful, but in some cases we might face troubles with the distribution that we use. When we have very few phenotypes and very large effects, we are going to end up with a very large test statistics. And this using chi-square distribution might be a problem because our p-values are going to tend to 0. Okay, so even it's not going to be possible to continue with further analysis, for example, even to correct these p-values, because everything's going to be 0. So how are you going to correct something that is literally 0 or approach very, very, very close to 0? So when you have these kind of situations, or even if you don't want to base your results on a chi-square distribution, there are alternatives, okay? Currently, we are developing studying where we are comparing different algorithms and different methodologies to estimate the pleiotropic effect in different scenarios. So scenarios, as I mentioned, with few markers, large effects, a large number of markers with small effects. And we are comparing the multi-trait statistics from Bolomar, methodology that is called CP-based, another one that is called PleioSNP that is based on permutations, and takes that use an independent association study that combines all the effects, not the summary statistics, but combines the effects during the association process, okay? Another point, when you do a GWAS or a multiple testing, we need to adjust adjust our p-values for multiple testing. And why we need to do this? Basically because when we set alpha, when you set a significance level, we are actually assuming a level of false positive, a frequency of false positive that we could assume, a type 1 error. So using a p-value of 0.05, we assume that we can have up to 5% of false positives, okay? And when you do subsequently association tests, we accumulate exponentially the probability of a false positive. So we need to adjust our alpha, our significance level, to avoid false positives. One very common approach that we see is the Bonferroni multiple test adjustment. What we do, we take alpha and divide by n the number of tests. This study, the theory per se tends to be extremely conservative, and I'm not a very big fan, even more after I have some long talks with Professor Flavio about Bonferroni and FDR and other correction tests, okay? But another thing, Bonferroni assumed that all tests are independent. And when you talk about genetics, we know that not all the tests in a GWAS are independent. Why? Because we don't have independent segregation in all the regions of the genome. I already mentioned haplotype blocks that are generated by recombination and linkage disequilibrium, okay? And also, when we talk about different chromosomes, we have a tendency of independent segregation of the markers in each chromosome. So the markers in different chromosomes tend to segregate independently, so the tests between chromosomes are independent, but within chromosomes, we might have the haplotype blocks that broke this independence of the tests, okay? Some ways to try to reduce this conservative uh, uh, pattern in the Bonferroni corrections is try to find the number of independent segregation blocks in the genome. There are multiple strategies actually, but on GALLO we implemented two. One that's called Simple M, another one that we're calling NSEG CHR. Simple M, we use the decomposition of square linkage disequilibrium matrices. So we estimate the eigenvalues from this correlation matrix, and we set a threshold. So once we reach, for example, 9.99% of the variance explained by this eigenvalue vector, we define this point as the number of independent markers in our analysis. And this can be done, for example, in each chromosome individually. And then we define the number of independent markers per chromosome. The other one is based on the number of independent segments approach that was described by Goddard and Hayes. And what we do using the effective population size, the chromosome length, and the linkage equilibrium, we try to estimate the number of uh, independent segments in each chromosome. And with this, you can adjust your number of independent tests and use this to apply a Bonferroni correction, for example. Okay. Just to finish, we also have functions to estimate gene level p-values. We are three approaches: weighted z-score, meta-analysis with linkage disequilibrium correlation coefficients, and the Davis algorithm. The Davis algorithm is the one that I'm seeing that performs better, and I described the approach in the last presentation, so I'm going to skip it now. But then you can have, for example, a Manhattan plot that shows a gene level p-value and not a marker level p-value, and this can be a very nice approach to try to find potential functional candidate genes. And we implement graphical visualization of connections using network approach, not the relationship, the port plots. And we did this using the Vis network package on GALLO. And also the possibility to extract centrality metrics from these networks. So for example, you have a very large network composed by markers and KTLs, and you can subset this network by, for example, only milk-related KTLs, and try to find the most central SNPs connected with the KTLs, the most central genes, and try to find the ones that could be more relevant for your process. And you have multiple centrality metrics implemented on GALLO in this version that we have on CRUN now. These approaches were already used in a paper that we published recently, where we integrate results of gene and CGIL annotation to try to find candidate regions across selection sweeps in the sheep genome. Okay, and why 2.0? Just to finish, because we were trying to implement all the studies that I show in my previous presentation about representative learning on GALLO. So we are doing these studies of pleiotropic effect, of different metrics for gene level p-value estimation to implement this representative learning pipeline on GALLO. It's going to be a big jump on GALLO because we're going to implement a model of gene prioritization, and this needs to be done carefully. This is why we didn't release yet, but this is going to be a big jump on GALLO. This is why we're planning to call GALLO 2.0. Just to finish, these are the GALLO repositories, the official ones. The CRUN official repository of R, my GitHub, that needs to be updated more frequently, and the GALLO vignette as well. The GALLO vignette still don't have the new functions, but I'm planning to have this in the next 2 months as well. So I'm gonna update the tutorial of GALLO. And just before, thank you all. If you know someone that might be interesting, we are searching a PhD student that's gonna join a project that will be a co-IP in the new institution that I will be joining the next month as researcher, and we're going to work with inflammatory challenges and resilience and try to integrate omics data, pretty much transcriptomics and metabolomics, to find candidate genes for higher resilience standards in dairy sheep. Okay. And thank you all. If you have any kind of questions or comments, I will be more than glad to answer with a very fresh photo of our team here in León that was taken today in the celebration of retirement of Dr. Fernando de la Fuente. Thank you very much.
Thank you, Dr. Fonseca, for your presentation. We are very glad that you were able to come back and tell us more about your work and the updates with GALLO. I'll open it to the floor for any questions. Anyone in the room or online, if you have any questions, that'd be great.
Pablo.
Thanks, Lucas.
Hey, Pablo, nice presentation. I got a little bit of the end there. I didn't get it quite clearly, actually. Is GALLO going to in the next update? Is GALLO going to have the use of independent segments of the chromosomes being used for the analyses as well, or we're not having that yet?
Actually, these 6 functions that I show in the table during the presentation are already in the GALLO 1.5 version. So they are already implemented on GALLO. So if you update your GALLO in your R version, you're going to have these functions already, okay? The functions that are not implemented already are the ones associated with the representative learning pipeline.
Okay. Awesome. Thank you.
You're welcome. I'm sorry, I don't know why my camera was off.
You're muted, Gabby.
Thank you, Lucas. So sorry, what I was saying was we are a few minutes over time. So if anybody does have any questions, feel free to reach out to Dr. Fonseca. But thank you again for presenting to us. Well, we will have the recording posted to YouTube again and send you those details later. But thank you everybody for coming, and we'll see you again next week.
Thank you very much, Gabriela. Thank you very much, everyone, for the opportunity to show GALLO today here. And if someone have any question, you can reach me out by email or anything, and I will be more than glad to talk with you guys.
Thank you.
Have a nice weekend.
You too.